DNA dietary analysis is a non-invasive tool to identify the food consumed by vertebrates. This is achieved by identifying prey DNA in their scats, which is especially useful for seabirds as feeding events are at sea and cannot easily be observed. Fortunately, during the breeding season, seabirds must return to land to raise young, making them more accessible to researchers. During this time, they are very good at projecting diet data around their colonies and nest sites in the form of poo. These colonies can be remote, often making them difficult and expensive to access. So we want to ensure that the samples we collect are of high quality and contain sufficient diet information, as returning to these sites again often isn't possible. This video provides field collection protocols for seabird scats, which were developed and optimised using shy albatross in Australia. These findings are likely to be relevant not only to other seabird diet studies, but to that of vertebrates in general. A fairly simple set of equipment is required for collecting scats in the field. You will need label vials with 70 to 80% ethanol. We use 2 ml vials for this. Straws or tweezers to collect the scat, bearing in mind that scats can be very runny, so something that can scoop is important. Also, you need to be able to clean it or replace it between collections. Kim wipes are a useful cleaning item in your kit. And finally, a notebook and pencil to record the sample number and any other desired information. When a fresh scat is seen, watch where it landed and approach slowly. Collect the dark part of the scat, avoiding the white, which is primarily urea and doesn't contain dietary information. Only a small amount of the scat is required, equivalent to half the size of a baked bean. But if the scat is large and solid, collect a small amount from multiple parts to ensure no biases occur. Place the sample in a vial and shake it hard to mix it with the ethanol. Keep the samples in a cool location out of the sun during the field trip, and place them in a freezer when you return to civilization. There are many considerations to make before commencing a study using DNA methods, and these will largely depend on the aims of the study. You will need to consider which genetic markers or primer sets you will use to address these aims and determine how and when the sample should be collected. Broadly speaking, 2 approaches are used for seabird diet analysis. Group-specific primers provide high taxonomic resolution of a specific prey group, such as fish or cephalopods. However, they don't allow all prey items to be detected. If this is required, a universal primer is needed to identify all food groups. However, this primer identifies all eukaryotes, which means that non-food DNA is also detected. Seabird scats contain the food DNA you are targeting, but also DNA from the consumer, from parasites, and unicellular organisms found in the gut. External contamination may come from the vegetation or dirt the sample landed on, insects that might feed on the sample or lay eggs in the scat, and there is potential handling contamination. Using shy albatross scat samples, we tested how to minimise the collection of non-food DNA in the sample and increase the detection of food DNA using universal primers. The following variables were explored: sample freshness, collection substrate, breeding stage, and the developmental stage of the bird, for example, chicks or adults. Seabird colonies are far from ideal lab conditions, with strong winds, heavy rain, and in the higher latitudes blizzards being common occurrences. These exposed sites also experience high levels of UV. All of these factors can degrade the DNA in scat samples. Our study found that fresh samples where the bird is seen defecating enable the highest detection of food DNA. To collect these for, say, burrowing seabirds, fresh scats might be obtained by laying cardboard or cloth outside the burrow. For surface nesting seabirds, find a location on the periphery of a colony and quietly sit, watch, and listen. Once you've been watching for a while, you'll start to observe distinct behaviours prior to defecation. These include the bird standing, shuffling around, usually to face into the wind, before they lean forward slightly to poo. This is usually followed by a tail shake. If you see any of these behaviours, be ready. Sometimes you will hear the poo, so quickly look for the bird shaking their tail and the direction their bottom is facing to be able to find the scat. Our study found that some recent samples that were still wet, but the deed hadn't been seen, still contained some food DNA, but larger sample sizes would be needed. In such cases, look for wet samples. Bubbles on the scat are a good indication that it is fairly fresh. Avoid any dry samples or those that are already forming a skin. these samples were found to be more degraded and contained more contamination from insects and fungal growth. Therefore, the amount of food DNA was minimal. Seabird colonies are often surrounded by tussock grass, which contains unwanted DNA that can contaminate the sample. Around nest sites, there is often exposed dirt which can contain unicellular organisms. We found that collection from rocks surfaces yielded the best results as there was minimal contamination from other DNA. If you are collecting samples from dirt or vegetation, try and minimise the collection of soil or plant in the sample. During incubation, birds are fasting for extended periods at the nest, with minimal food DNA in their scats. We found that scats from birds that have been back at their nest for greater than 24 hours contain almost exclusively parasite and bird DNA. To obtain diet information during this period, target birds that have been back at the colony less than 24 hours. This may involve watching birds that are changing incubation duty and monitoring that nest over the following day. Or for some species, waiting at known access routes and watching birds as they return from sea might be the best option. Alternatively, if ethics allows, marking a bird with stock marker may be a way to track when birds swap over, like the bird on the right-hand side of the screen here. Overall, where possible, avoid any birds known to have been fasting. This includes those building and defending nests. The colour of the plumage can also be a good indication of how long the bird has been ashore. In our study on shy albatross, scats from young chicks still being brooded had less food than older chicks or adults. Instead, samples had a high proportion of consumer or bird DNA. Food is likely to be partially digested prior to feeding by regurgitation, and parental DNA is also likely to be transferred, which may explain the high bird DNA proportions found. If collections during brood are required, target breeding adults rather than small chicks. If samples from chicks are required, wait until later in the season when they are older and larger, as the food DNA proportions were much better then. In summary, to get higher quality diet data from scat samples, try and collect fresh scats where the bird is seen defecating. Be mindful of contamination and minimise the collection of dirt or vegetation in the sample. Collect scats from animals with minimal time since feeding and avoid fasting animals. Avoid young chicks, instead target larger, older individuals or adults. And finally, keep collection protocols consistent between sites so that any dietary data that you obtain can be comparable. Hopefully this short video has been very useful, and now you can go out and thoroughly enjoy your scat collections.